Hello, welcome back guys in another video from microbiology with Shagnik. So today we will going to talk about a important part of our electron transport chain, that is chemiosmotic theory. So the chemiosmotic theory is a theory that was proposed by Peter Mitchell to explain the idea how energy released during the respiration is used up for the synthesis of ATP. Because during the discussion of our electron transport chain, we have talked about the energy or free energy, right? That was released, right? So there was a question how enzyme ATP synthase, which will going to play an important role in synthesis of ATP, will going to use this energy, okay, and synthesize ATP from ADP. So Peter Mitchell used this chemiosmotic theory to answer this question. So what is the question? So here we will try to find out the actual mechanism by which energy released from the respiration will be used up for the formation of ATP. So in the year 1961, Peter Mitchell proposed a mechanism called chemiosmotic coupling to answer this question. So here I have written the entire thing. So first take a screenshot of it so that you can use it to make a note for yourself. So then let's start with the diagram to understand what is chemiosmotic theory or chemiosmotic coupling theory. So here you can see it is mitochondria, right? This is the matrix, this is inner membrane space, and this is outside. Okay. Now what is the idea? The idea is during the electron transport chain we have talked that there will be release of energy, right? Now the energy or the free energy that is released will be used to drive a active transport system, okay, and this active transport is the transfer of proton from the matrix to the inner membrane space. As you can see, protons are transferring here, not only particularly in this place but everywhere, okay, throughout the inner membrane space. Okay, so now again I am repeating what is going on, that the energy or free energy released during the electron transport chain will be used up for a active transport system, that is transfer of proton from the matrix to the inner membrane space. Clear? And as a result, what will happen? It will create a transmembrane proton gradient, right? Because as you can see, first of all, remember one thing very carefully, that increased concentration of H plus decreases the pH, and increasing concentration of OH minus or decreasing concentration of H plus will going to increase the pH. Okay, so that is the idea. So as in the inner membrane space the concentration of protons are increasing, it will surely going to decrease the pH, right? And as a result it will create a electrochemical gradient or pH gradient, whatever you say. Okay, and now under such circumstances there will be a thermodynamic tendency of those protons present in the intermembrane space. What they will try to do? They will try to go back inside the matrix, okay, so that the pH of matrix and the pH of the intermembrane space becomes same. Okay, now try to think about this. We have used energy to transfer protons from matrix to the inner membrane space, right? Now those protons are trying to enter inside the matrix, right? So it is obvious that during that comeback process there will be release of heat or dissipation of heat. Sorry, what I am telling, there will be dissipation of energy, right? Because here we have used the energy for transferring it from matrix to the inner membrane space. So obviously when it will try to return, there will be release of energy, right? Now the energy that is released during the comeback process of protons present in the inner membrane space, this energy, not all the energy but some of the energy from those energy will be used up by the ATP synthase for the formation of ATP. That is what chemiosmotic theory wants to tell us. Okay, so that is the idea. Again, for a quick recap, the electron transport chain releases energy, right? And now this energy will be used up for a active transport system. Okay, and what kind of transport it is? It is the transfer of proton from the matrix to the inner mitochondrial space, right? Or you can say intermembrane space. Okay, and as a result, what will happen? The concentration of the proton will be increased in the intermembrane space. That will decrease the pH of the intermembrane space, right? And as a result, there will be a electrochemical gradient that is created. So the thermodynamical tendency of those protons will be to come back inside the matrix, so that the pH of the matrix as well as the pH of the inner membrane space becomes same, right? So as a result, what will happen? That is, we have used the energy of electron transport chain to transport the proton from matrix to the intermembrane space. Now those protons present in the intermembrane space tries to come back in the matrix, so the energy will be released, right? And those kind of energy will be used up by ATP synthase for the synthesis of ATP. So that is the idea of our chemiosmotic theory proposed by Peter Mitchell in the year 1961. Now the question is there any experimental procedure or is there any experiment available to prove this theory? Obviously. So let's see the experiment done by scientists Andre Jagan and Ernest Urib in the year 1966. So what they have done? So first take a screenshot of it so you can use this to make a note for yourself. Okay, I hope you have taken this. So then also take the note, a screenshot of this 2 point also. So now let's move on the part, that is the experimental proof of chemiosmotic theory done by Andre Jargon and Ernest Rip. So here what they have done, they have equilibrated chloroplast thylakoid vesicles containing CF0 CF1 particles. What they have done, they have equilibrated chloroplast thylakoid CF0 CF1 containing vesicles. Okay, so what is CF0 CF1 particles in the thylakoid vesicle? We will going to talk about this in some other videos. So now the idea is they have equilibrated it in a buffer with pH 4 in the dark condition, that is there will be no presence of light. Okay, so now here first the actual intention was to make the pH of the lumen, which was initially pH 7 to pH 4, because remember the pH of the lumen and the pH of the environment try to maintain a equilibrium. Okay, and so, and for doing this, if you going to isolate, so if you equilibrate a chloroplast thylakoid in a buffer of pH 4 under dark environment, the lumen of the or the pH of the lumen becomes same of the solution where you have equilibrated it, meaning that after some time the pH of the lumen becomes 4. Okay, so what they have done, if we try to start it again, they have equilibrated a chloroplast thylakoid vesicle containing CF0 CF1 particle in a buffer solution of pH 4 in the dark condition, right? And after some time the lumen of the pH of the lumen of the chloroplast thylakoid vesicle becomes 4, right? That is the equilibrium with the environment. Now what they have done, they have rapidly transferred this thylakoid vesicle in a pH of 8 containing buffer solution filled with ADP and PI. Okay, and remember again, here also the dark condition is mentioned. Okay, so as a result, what will happen? After some time, it was observed that ATP is getting synthesized. Okay, now the question is, here we have not used any kind of energy, because as here we have not used any kind of light, there will be no form of energy here. Okay, so how this ATP is generated. So after experiments, it was observed that the transmembrane proton gradient that facilitates the removal of proton from the lumen in the environment also drives the synthesis of ATP. Okay, so that tells us that yes, transmembrane proton gradient or electrochemical gradient or pH gradient have the ability to synthesize ATP. That is what chemiosmotic theory wants to tell us, right? So that is the idea. So for a quick recap, scientists Andrei Jagendrov and Ernest Reeb in the year 1966 proposed a experiment to prove the chemiosmotic theory. What they have done, they have equilibrated a chloroplast thylakoid vesicles containing CF0 CF1 particle in a buffer of pH 4. Okay, what was their initial intention to do this? Their initial intention was to drop the pH of the lumen to 4. Okay, because as we have just discussed that there will be a tendency to maintain the equilibrium with the environment, right? So the pH of the lumen becomes 4. Now once the pH of the lumen becomes 4, the chloroplast vesicle will be immediately transferred to a jar or to a system containing a buffer solution of pH 8 filled with ADP and PI. And again, here also the dark condition is maintained. Okay, so now after some time it was observed that ATP is synthesized. So the idea is, as here we have not provided any kind of light, so the energy which is driving the synthesis of ATP is none other than the transmembrane proton gradient, right? That facilitates the movement of proton from lumen to the buffer environment as well as facilitates the synthesis of ATP. Okay, so this is not a only experiment to prove the chemiosmotic theory. There was other experiment which was similar as this one. So here inside out preparation of sub mitochondrial vesicles was used to create a artificial membrane electric potential for ATP synthesis. Okay, so you may remember only one, that is the Andrei Jagendrov and Ernest Urib in 1966, and it was very easy to remember. So that is the idea of chemiosmotic hypothesis and also the experimental proof of chemiosmotic hypothesis. So I hope this video will be helpful for you. Thank you.